This initial research attempts to explain fish's feeding patterns based on the stimulation of olfactory receptors. With fish being primitive organisms and having a primitive sense of smell, they make excellent research organisms. A fish's sense of smell is very different from our perception of smell in that the sense of smell is more like a sense for the presence of food. Here we can see the fish's brain and the site for olfactory reception. These olfactory chemoreceptors found on the olfactory bulb open and close in response to specific concentrations of amino acids, which will change based on the presence or absence of food. This food profile, or the availability of specific amino acids, can and will vary. With the goal of stimulating fish feeding, it is important to understand which profile best stimulates fish. In your typical fish meal, there are higher concentrations of charged amino acids compared to uncharged, which tend to occur in meatier foods, while higher concentrations of uncharged amino acids tends to occur in more vegetable-based food sources like soy meal. This uneven distribution of charged versus uncharged amino acids is the driving force that stimulates fish to feed. These primitive olfactory receptors detect the presence of food by binding to these amino acids, and certain profiles are more stimulatory than others. Interestingly, each olfactory receptor protein has two binding sites, one for neutral amino acids and one for charged amino acids. What has been observed is when a neutral amino acid concentration is higher compared to charged amino acids, more neutral amino acids bind to the available lone receptors, meaning these sites are unbound by a charged receptor. And then this leads to a stimulatory response, causing fish to search for food. As you can see from the animation, binding of a neutral amino acid stimulates a response illustrated by the protein channel opening and allowing sodium to enter into the cell membrane, which results in an action potential and hence stimulation. Similarly, if the concentration is higher of charged amino acids, this too will result in stimulation as charged amino acids are binding to that lone charged receptor site. Surprisingly, when concentrations of charged and uncharged amino acids are equal, stimulation ceases to exist as modeled by the animation here. Simply put, only one type of amino acid, either charged or uncharged, can be bound to the olfactory protein receptor and result in fish starting to search for food. Knowing that stimulation occurs via high concentrations of either charged or uncharged amino acids, the ultimate goal is to elicit a response by reaching appropriate stimulatory mixtures. Again, having relatively equal concentrations of charged and uncharged amino acids has a canceling effect.
My name is Chris Hanneman. I'm a chemistry major and I'm researching under the Valine Group.
My name is Emily Hankins. I'm a chemical engineering major. Next fall I'm going to be transferring to UIC to pursue a degree in chemical engineering, kind of with a more biomolecular focus, and I'm hoping to eventually get my PhD from UIC. I'm part of the lysine group, so my partner Nicole and I, we run the experiments, we collect the data in our journal, we kind of make a catalog of what we did that day, and we film the experiments and kind of look over the films, collect more data.
My name is Sean Odell and I'm majoring in biochemistry. So my role is to assist in research procedure itself, as well as create content for our social media platforms like our Facebook page to communicate our research to the public.
My name is Nicole Quillis and I have a bachelor's in psychology from UIC, and I'm planning on becoming a pharmacist. I am specifically looking at the effects of lysine on fish feeding behavior.
My name is Daniel Shaligo. Currently I major in biology and I plan to become a professor at a university. I'm the group statistician. I collect all the data from our labs and I analyze it.
My name is Stephen Blazek. I'm a nursing student and I also work with 3D animation and video production. I do research on what exactly we're doing, what parts of the fish anatomy that we're affecting, and so I try to create 3D models and simulations of how exactly all of that works.
My name is Janelle Sariaga and I'm a biochemistry major. I work with Team Valine and I basically help them run the experiments.
NSCI 296 is an opportunity for students to get undergraduate research experience. There are 7 of us students in the group. We're working under Dr. Mills and Professor Czerniak, and every single student has a specialty that they work with. And so we're working with fish to try to get them to feed involuntarily.
We have 2 filters in the tank. We have a canister, which is submersed, and we have a hang-on-the-back filter. And every week we change the activated charcoal out of there, And every 2 weeks, we scrub the tank walls and do a rather large water change. Basic day of research involves us getting to the lab at about 8 in the morning and deciding on what we want to experiment with today. So say we pick a solution that we want to experiment with. We will first make up the solution in the lab, and then we will shut off any bubblers or filters that might interfere with the dispersion of the amino acids. After the tank is prepared, we load our cartridge with a point source of food and we place it into the middle of the tank right under our jet stream, but above our silicone bowl, and we let it rest there for about 10 minutes so the food can saturate and the fish can recognize that there is a food source present. After 10 minutes of resting, we dose the cartridge with about 50 milliliters of amino acids. We record the data for about 25 minutes and we analyze that at a later date. So as soon as the cartridge is in the water and the amino acid is administered, it's just a waiting game.
So the importance of data analysis lies upon the fact that you need to build evidence for your claim. We want to prove that fish feed more with amino acids versus no amino acid. So we take a period of time away from the video at first in order to give a control. We don't want the recording of the data to be of the fish being introduced to something new, like the sponge cage. Then we record a 10-minute segment, and in that 10-minute segment, we look for pecks and we look for periods of activity. A peck can be two different things. It's either when the fish drops down, pecks a rock, and comes back up, or when the fish is already down, moves back, we count to 2 seconds, and then it goes back to pecking. Periods of activity consist of 30-second intervals that we count how many fish are in the area of activity, and through that, over the whole 10-minute period of time, we can collect an average of how many fish are in the period of activity. We hope that the amino acids show greater pecks and greater activity periods than no amino acids.
Last year's research group conducted a dye experiment, and this year we are building upon that and repeating the dye experiment in order to optimize the release method of 2 different amino acids, both lysine and valine.
The food experiments involve testing different food sources for the fish to focus on, so you could call them targets. Once the amino acids hit the fish's receptors, you need something for the fish to focus on and be drawn towards. This means that you don't want a food that is too overstimulatory or too understimulatory. We've tried things from horse pellets to carp pellets to even lifesavers to try to get this effect. We're finding that sweet feed, horse sweet feed, works best, creates just enough of a response.
We first conducted a kind of preliminary lifesaver test. We put the lifesavers in the bottom of the tank and just watched the fish. We discovered they weren't really stimulated by these lifesavers. Later in our research, we discovered that sugar can produce a fear response in some fish, and we concluded from that that maybe the synthetic sugary lifesavers was not the point food source we should be using.
The future of the project, we're looking towards getting this research done at a more serious academic level. So right now we're starting at a community college level, and then right now we're thinking about transferring towards more serious university type research. Just like the groups before us have done, we're trying to set up parameters as to how the experiment should be done, and also we're going to work on improvements as to what we can improve with the experiment itself so that other groups in the future can replicate it.